I'm using chemistry vodka because my friend Kate Hudson introduced it to me. And she also introduced me into how...
For those of you who aren't muted, please mute yourselves. Please remember to mute yourselves. Okay, so I assume now that everybody has brought a pencil and paper, a pen and paper. So what I would like you to do is to draw a carbon atom. Just draw C. And on each side of the C, you will draw the hydrogen ion. You will draw the carboxylic group, and you will draw the amino group. Okay, I'm assuming all of you are doing this. So here we have an amino acid where we have a carbon atom, which has all these four groups, which basically saturates this carbon atom, right? So we have the amino group, we have the carboxyl group, we have the hydrogen, and we have a side chain, which is called R here. So this side chain can differ from amino acid to amino acid, which categorizes the amino acids into many different classes. For example, we have polar amino acids, we have non-polar amino acids, we have charged amino acids, we have neutral amino acids, etc. So all of these amino acids are defined by the side chain, which means that every single amino acid has this amino group and it has the carboxylic group. So when a peptide bond is being generated, and this peptide bond is generated during protein synthesis that we will learn a few weeks later, this protein synthesis, as you know, is happening with the help of our ribosomes by a process called translation. So when translation is happening and proteins, the peptides are being synthesized, this peptide bond is formed between two amino acids. So I would like you, please bear with me and try to draw this particular picture that I am pointing at with my cursor, and draw it in a piece of paper, in your book, your notebook that you have in front of you. I am drawing it as well, because drawing it will help you to understand how the bonds are formed and how the water is being released. So just as we learned for carbohydrates, when two glucose molecules join together by what is known as a condensation reaction or a dehydration reaction, it results in the release of a molecule of water. In the case of lipids, when the glycerol molecule, the OH of the glycerol molecule combined with the carboxylic group of the fatty acid chain, again, it resulted in the release of water. In the case of fatty acid, in the case of triglyceride, 3 molecules of water are released. In the case of phosphodiester bonds that we learned yesterday, when nucleic acids are joined together, nucleotides are joined together, again, it results in the release of a molecule of water, as well as in the case of the nucleotides, a pyrophosphate, right? So this dehydration or condensation reaction is common to all polymerization reactions that we have in our living cells. And this is common to all life forms, starting from bacteria all the way to larger life forms, okay? So by now, you will have drawn this figure. So please remember to label it correctly, R and the N terminus and the C terminus, etc. So you have the OH of one amino acid, the hydrogen from the N terminus from the nitrogen of the next amino acid, giving us what is known as a peptide bond with the release of a molecule of water. So water is being released and we end up with NCC, NCC, OH, N already has the NH2 group. And then we have the R's, don't forget to label the R's. The correct carbon should have the side chain and the oxygens. So make sure you label where the peptide bond is. Now, if you have already done this, then you will realize that what we have in this peptide bond, in this tiny peptide consisting of 2 amino acids, is something called an NCC. NCC. So this is at the backbone, literally, of the peptide chain. I want you to remember this NCC because it will become useful for you to understand how the secondary structures are formed when we come to that part of the lecture. One other thing that I would like you to notice is, for example, if there was another peptide bond formed downstream of this peptide bond and another, you would end up with one end of the peptide bond having an N terminus, having the nitrogen, and the other end of that peptide having a carbon. So we have a direction in a peptide, right? We have an N terminus and we have a C terminus. And when you talk about proteins with their N termini and their C termini, this is exactly what it means. It means that one end of the amino acid right at the first amino acid of that protein, the N terminus is called, the nitrogen is called the N terminus, and the carbon of the last amino acid of that protein gives us the C terminus, okay? All right, there is a question, let me open it. The amino group should always be yes. I mean, that's how we are writing it. So the question is the amino group always on the left and the carboxyl group on the right? Absolutely right. It's the way we are writing it in the book gives it a left and right, but yes, the peptide starts with the N terminus and it ends with the C terminus. Okay, so the first amino acid always has the N terminus, gives us the N terminus, and the last amino acid gives us the C terminus. What this also means is that the N terminus is the start of the protein, the C terminus is the end of the protein. Okay, so the next slides are just larger versions of what we have seen. So here we have the side chain, we have the amino group, we have the carboxyl group that we've just learned, and I hope all of you have had some practice in drawing how the peptide bond is formed, how the water is released, and this is the peptide bond. And again, I'll remind you, please have in mind this NCC sequence that we are seeing here. What is the alpha sign on the carbon in the middle? Okay, so the alpha sign that we have here is indicating that this carbon is bound to 4 completely different groups. So they are not identical to each other. That's what is that is what is signified by the alpha. So again, here you can see it. The R can be a different group, the C can be a different group, it is a different group, the N is a different group. So the carbon is attached to 4 completely different groups is what it signifies. Okay. All right, so now I am going to go, I'll stop sharing for a moment, and I'm going to go to our videos. So here I'm going to start again with the Smart Biology book, and we will start with the amino acids. So let's have a look at what we can learn about the amino acids. So amino acids, as we just learned, they have a central carbon. And you can see it here. They have a backbone consisting of a carboxylic group that we've just seen, an amino group, and then we have the R group. The carboxylic groups are acidic, therefore it's called an amino acid. So the side group defines what amino acid we are dealing with. And as you can see, there are 20 different amino acids that are found. So these 20 amino acids are used for protein synthesis. That doesn't mean that we only have 20 amino acids. We have some unusual amino acids in our body as well. Citrulline, for example, as far as I know, is an example of an unusual amino acid, which is not utilized for protein synthesis, but it's called an amino acid because it has a carboxylic group and it has an amino group. There are other examples of the unusual amino acids as well. As far as I remember, citrulline is one of them. We'll need to check it. But the 20 amino acids that are used for protein synthesis, they fall into several different categories that we will see in a minute. And these categories that they fall into will define what their roles will be in the cell. So it's not clear why 20 amino acids have been selected, but these have been selected billions of years ago because bacteria use the same amino acids in order to generate their proteins as well. So there could be other chemical side groups which could function in a similar manner, but nonetheless, we have these 20 amino acids. Okay? So they are used in all organisms from bacteria to humans, and they have a wide range of chemical side groups. So the R group is what gives us the characteristics. So they can be positively charged, and here the positive means that it has a positive charge at physiological pH, a negative charge at physiological pH, which is pH 7.4. Some of them are polar, which means that they are soluble in water. So serine, threonine, tyrosine, all three of them have hydroxyl groups to them. Serine, threonine, and tyrosine are the three amino acids that can be phosphorylated. These are the only three amino acids that can be phosphorylated. So they are enormously important in helping us understand how proteins can get phosphorylated. And we're going to be talking about phosphorylation of proteins a lot in the next few weeks. But these are the three amino acids that can get phosphorylated via their hydroxyl groups. Cysteine is a very interesting amino acid because it has a sulfur group to it. And this sulfur allows the cysteine, two cysteine molecules to join together in a covalent bond, forming what is known as a disulfide bridge. We will look at that as well later. And then we have the hydrophobic amino acids. These hydrophobic amino acids tend to have a benzene ring-like structure in them that you can see for the phenylalanine here. And this, the hydrophobicity of these amino acids allow them to be incorporated, for example, into our membrane. So if we did not have the hydrophobic amino acids, we would not be able to have membrane-bound proteins. So as we just learned, so please do the interactive exercise for those of you who have the book. So amino acids can also exist as optical isomers because the carbon is joined to 4 different groups, and then they can form these identical mirror images of each other. So we have the L isomer and we have the D isomer, but amino acids, unlike glucose, which is only found in the D isomer, amino acids are found in the L isomer. So we only have L amino acids and we only have D glucose. But exactly why these were selected by evolution, again, is something that has not been defined yet. Okay. So again, now we are going to look at the formation of the peptide bond. So this is tryptophan, and this is tryptophan that can be converted into serotonin. Okay, so let's go back just a couple of seconds. And here, basically, the monomer itself, this is the amino acid itself, which can function as a precursor for the synthesis of a very, very important neurotransmitter. Serotonin is a very important neurotransmitter, and it is synthesized from tryptophan. So tryptophan, remember, was one of our highly hydrophobic amino acids, and it can be converted. You can see from the structure where the changes are. So we have a hydroxyl group over here, I think. So these monomers can act as precursors for the synthesis of important biological molecules. For example, tryptophan is used to produce serotonin. And there are many, many examples of individual amino acids being used to synthesize neurotransmitters, to synthesize hormones. For example, the thyroid hormone is also synthesized from amino acid precursor, norepinephrine, epinephrine, these are all synthesized from amino acid precursors. They can also, of course, be polymerized into polypeptide chains. So now we are going to look at the polypeptide chains, whereby they will be polymerized, forming proteins with very complex 3-dimensional shapes. So this complexity, this 3D shape of the protein, as you all know very well, gives the protein their function. The shape of the protein is absolutely critical in their function. And the structure and function of each protein, therefore, depends on the amino acid sequence. Because if you have a chain of hydrophobic amino acids in the protein, those hydrophobic amino acids will tend to clump together, right? They will want to be together because like attracts like. So two hydrophobic amino acids will come closer to each other, forming hydrophobic bonds. Whereas if you consider a three-dimensional structure of the protein, the outside amino acids, the amino acids that are found on the surface of the protein, are all expected to be polar amino acids. Can anybody tell me why we expect polar amino acids on the outside of the three-dimensional structure of the protein, on the surface of the protein? You can open your microphone, or preferably you can write in the chat. Why are polar amino acids expected to be seen on the surface of the three-dimensional structure of a protein?
Can I say something?
Yeah.
Because they will interact with water, and there will be also a hydrophilic interactions between the atoms inside the protein polypeptide chain, because they get together and cluster.
Excellent. Yes, absolutely correct. So the answer is that the amino acids that are on the surface of the protein are the amino acids that will interact with the hydrophilic environment. So we learned this yesterday and last week as well, that the inside of the cell is highly hydrophilic, isn't it? It's a watery environment. Outside of the cell is also highly hydrophilic. It's a very watery environment. So therefore, it's expected, it's absolutely logical for the protein to have the hydrophilic amino acids to the surface, the polar amino acids to the surface, and the non-polar hydrophobic amino acids are clustered towards the inside of the protein structure. Okay, so let's continue now. So the specific structure is defined by the amino acid. So here we have a polypeptide chain which has folded a little bit. And as it folds, okay, let's come to the peptide bond now. Amino acids are joined together by condensation reaction. This is the reaction that you have drawn. So again, please mute yourself. So just like we learned with sugar molecules, and you learned a little bit a few minutes earlier how to draw the peptide bond. So they are joined by condensation reactions. So again, there will be a release of a molecule of water, and it will result in the formation of the peptide bond. So here we have the release of the water. This is the amino group, this is the carboxylic group. They're coming together, releasing a water and forming the peptide bond. So as we also learned a few minutes ago, every polypeptide chain has a carboxylic group at one end, and it will have an amino group at the other end. This gives the polymer a directionality and a structural polarity. Structural polarity meaning that there will be an N terminus of the protein, a C terminus of the protein, and this will define many, many different properties of the protein, as we will learn today and in the weeks to come. So we have the carboxylic group here, we have the amino group here, the N terminus and the C terminus. So now, the next few videos are really, really cool videos. I enjoyed watching them very much. We'll go through them, and they will tell us a little bit about how the protein structures are formed. So the different levels of formation of protein structure, starting from the primary structure to the secondary, to the tertiary, and then to the quaternary. So these are really, really interesting videos, and I encourage you to listen closely. So proteins, as we just saw, it's a long polymer of amino acids. It folds onto itself, producing a very, very specific three-dimensional shape. So here, mostly we represent, I mean, in this book, the carbons are represented by these gray circles and as ball and stick atomic models. But since the protein structure is very, very complex, they will simplify the picture a little bit. And let's have a look now. So this is how the protein would look if we considered the electron clouds around them. Okay, so the figures that we are drawing with the carbons and the hydrogens and the nitrogens, they are actually quite simplified, and it will be simplified even more where we will just see the NCC bond, the backbone of the peptide bond or peptide chain. But the entire structure of the protein does not quite look as simple as we represent them in the pictures, because they have, because we look at them, we need to look at them as a whole instead of individual atoms. So this is also a very interesting picture. So these are surface structure representations of these macromolecules. And here the blue outline that you saw represents the electron clouds around them. So I'm going to again pause the video for a moment, and I'm going to show you here in this picture how diverse the protein structures are. And I'm going to rewind and I'll show it to you again. Here we are seeing one protein that is sort of wound around another protein. So this is a complex structure of a protein. We have some proteins with more than one subunit. So you can see that there are 2 or 3 different subunits joined together. Here is a single polypeptide that has folded on itself to give us a protein. Here we have a membrane-bound protein. This is a receptor that is located in the membrane. And one thing that I will mention now, and I will mention it over and over and over again in the next few weeks, is the part of the protein that is located in the membrane is generally, it has a helical structure, okay? And this helical structure allows the hydrophobic amino acids to be in close contact with the fatty acid tails, okay? So keep that in mind as we go through the lecture, and you will appreciate it more and more as we go through the different weeks. So again, in this protein, this is a transmembrane protein, where again, you will have a part of the protein that is located embedded within the membrane. And obviously this part of the protein will have highly hydrophobic amino acids facing the fatty acid tails, okay? So here we have very diverse structures, and on the right here is one of our favorite proteins, and it will become your favorite protein since it looks so very, very amazing. This is a protein called PDH, pyruvate dehydrogenase. So and here you can see that there are many subunits. If you recall, yesterday we learned PDB stands for protein database, and these codes are will take you to the specific structure of the subunit of the pyruvate dehydrogenase. Pyruvate dehydrogenase is an enzyme that is found inside our mitochondria, and it is responsible for converting pyruvate into acetyl-CoA. It has many subunits, and it basically dances all the time while it's working. So let's have a look, and I'll rewind it again. Look at how amazing these proteins are. Okay, so this is how they are carrying out their specific functions. Okay, so this is our protein, and we just saw so this is the pyruvate dehydrogenase located within the mitochondrion. So just as amino acids are the building blocks, building blocks of proteins are the building blocks of our cells. So the proteins comprise about 25% of the cytosolic mass. The rest of it would be the membrane made up of lipids. We have a vast amount of carbohydrates that are stored, etc. And 50% of the mass of the cell membrane is also protein. So a huge, huge amount of protein is actually located on the membrane, constituting most of the dry mass of the cell. These proteins that are embedded in the membrane are synthesized in the endoplasmic reticulum. They are trafficked to the membrane, and they stay on the membrane in order to take signaling molecules in order to provide structural support as well as functional support, interaction with the extracellular matrix, and functions similar. So here we have, again, I'm going to pause the video, and you can again see a large number of membrane-bound proteins where they have a part of the protein that is looking towards the outside of the cell. So this is the extracellular space. Then we have a transmembrane domain, which is located embedded in the membrane. And then we have an intracellular domain, which is looking to the inside of the cell, where these proteins may potentially interact with other proteins in order to carry the signal inside. So here we can see that there are some proteins, and if you notice it carefully when I start the video again, there are some proteins that completely traverse the membrane. It's a completely, so you have one domain on the inside of the membrane, one domain on the outside of the membrane, right? So these are called transmembrane proteins. And then there are some proteins which are just associated with the membrane. They are not embedded in the membrane, but they are associated with the membrane. They are mostly found towards the inside of the cell, and they generally form a part of the cytoskeletal complex. They may not have any part of the protein embedded in the membrane, but they will have some part of the protein that will come very, very close to the phospholipid head groups, and they will remain attached to the inner surface of the membrane. Okay? And then there are some proteins which are anchored to the membrane. So these are called GPI anchors, which means that they have a little finger-like projection, which is made up of a glycophosphoinositide. So it's basically a glycoprotein, which not embeds, but it sort of sticks the protein inside the membrane. And we will see examples of those as well. So let's have a look now. So proteins are also, since they have very unique shapes, they give very specific shapes to the cell as well. So structural support to the cell. So for example, neurons and their very specific shape can be attributed to proteins as well as to the membrane lipids, of course. And red blood cells with their very distinctive shapes, very distinctive morphology, is also dependent highly on the proteins that are found in the membrane of the red blood cell. Proteins can catalyze chemical reactions. They can act as signaling molecules. For catalyzing chemical reactions, we can think of enzymes. For acting as signaling molecules, you can think of neurotransmitters, you can think of growth factors. They form the cytoskeleton, so you can think of the actin filaments, you can think of the microtubules, and they also coordinate the packaging of the DNA. In this case, we are talking about the histone proteins. Histone proteins, especially in eukaryotes, of course, allow the DNA to come very, very close and form the very distinctive higher order structure that we see in our chromosomes. At the end of this particular video, I'll give you a chance to ask me questions. So let me go back just a moment and show you this picture. In this particular video, you are going to see a molecule, a small molecule, which could be ATP, it could be a growth factor, they are generally very small signaling molecules, it could be calcium, which will bind to the protein and it will result in a change in the protein. So let's have a look. I'm going to go back just a little bit more because these are really cool. So here we saw a phosphorylation event. A protein can undergo many changes. Let's go back again. Let me slow it down a bit. So here we saw a phosphorylation event which resulted in a major alteration in protein structure. Here, this is even better. This, let me explain to you and then I'll turn on the video. This is the G protein coupled receptor signaling. So here is a protein called a G protein coupled receptor. This receptor, as the name suggests, receives signals from the outside of the cell. When it receives the signal from the outside of the cell, it signals to a G protein. And this G protein is capable of binding to GTP and getting activated only when it receives a signal from this G protein coupled receptor. This G protein is a membrane anchored protein that I just mentioned. It basically has a little finger-like projection that helps it to attach itself to the membrane. This is one of those. And then when it gets activated with the binding of a GTP, it will then move along the membrane because our membranes are quite fluid, so they allow the protein to move, and it will activate another protein, in this case, This is called something. This is something called protein kinase A. Okay, this is called adenylyl cyclase. Sorry. So when the G protein is activated, it will move and it will activate another protein called adenylyl cyclase, which will then result in downstream signaling. So I'm just going to go back a few seconds and show this to you again. Now it's a bit slower. So this is a phosphorylation event, which results in conformational changes in the protein. Or here we again have activation. This is the G protein that has now moved. This is the GPI anchor. It has now bound to adenylyl cyclase. This adenylyl cyclase, when it's active, will, let me pause it again, will result in the conversion of ATP to cyclic AMP, which will have various signaling functions. Again, I'm going to go back. So this is ATP being converted to cyclic AMP. And here, again, beautifully, we have actin polymerization. So this is a cytoskeletal protein that is getting polymerized in the cell in order to help the cell to move, perhaps. Proteins provide, again, shape to the DNA. So this is a tetranucleosome, for example. Here we have the microtubules, and on the microtubules, you can see also a number of motor proteins. These motor proteins help to move various different macromolecules on the surface. So here we have insulin, we have glucagon. These are also proteins. We have an antibody. This is an anti-canine lymphoma antibody. So these are all real proteins. Remember that, keep that in mind. This is the green fluorescent protein. This is the ATP synthase. So ATP synthase has many, many, many subunits. ATP synthase is also something that we are going to be talking about a lot, most likely next week, where ATP synthase is an enzyme that is located in the mitochondria, the inner mitochondrial membrane. And this ATP synthase is responsible for the conversion of ADP and inorganic phosphate into ATP. So let's have a look at how beautifully this particular enzyme works. So this is our antibody. And then let's go to this is the green fluorescent protein not found in higher organisms. And this is our ATP synthase. So it basically rotates in the membrane and it results in the formation of ATP. And this is our favorite protein, the pyruvate dehydrogenase complex, enormously huge complex of protein. It actually does look like this. And it has all these very nicely moving subunits, which are essential for the function. This is the 3D structure of the protein. Let's enjoy this picture for a moment. Okay, so now I'm going to give you a chance to ask me some questions. So there are several levels of organization within a folded protein. The first is, of course, the amino acid sequence. This sequence defines the protein. So every single protein is defined by its amino acid sequence. And that makes perfect sense because you can actually, once you know what the amino acid sequence of a protein is, we can predict. There are many prediction programs that are available on them, free to use, where you can enter the amino acid sequence and you can predict how this amino acid, how this protein will actually fold. And it ultimately determines the three-dimensional shape that the protein will fold into. These are different amino acids. You have histidine, glutamine, serine. Let me reduce the speed a little bit. So it represents the first level of structural organization, and it's called the primary structure of the protein. So now we will, the video will remove all the side chains, and we are just going to show it represented as green spheres. Okay, the side chains are there. The primary structure folds into two major ordered secondary structures, the alpha helices and the beta sheets. So you can see the alpha helix here, the beta sheet here, and you can understand that there are differences. So these secondary structures are formed by hydrogen bonds. So here we come to some of the bonds that are necessary for the formation of the secondary structure of a protein. Hydrogen bonds are the most important. Hydrogen bonds are relatively weak, but then when you have many of the hydrogen bonds, it becomes a relatively strong collective bond, right? Just like we have in DNA, two strands are joined by hydrogen bonds. Similarly, the secondary structure of proteins are also joined by hydrogen bonds. And here, the hydrogen bond is between the NH group and the CO group in the backbone. I would like you to very, very quickly steal a glance, have a look at the peptide bond that you have just drawn at the beginning of the class. So you have an idea where the NH and the CO groups are. And you can then understand how the hydrogen bonds are formed. So these NH and CO groups in the backbone of the polypeptide chain, and every amino acid in every protein, with the exception of one amino acid called proline, contains these same groups at the same position. So where the NH is and the CO is does not change, because remember I mentioned at the beginning of the class, please recall this NCC backbone, and you can see that from the picture that you've drawn as well. This NCC backbone remains unchanged in all amino acids except proline. So here we have the NH group, here we have the CO group. So you can see the NH and the CO, and the hydrogen bond will be formed between the NH and the CO of the fourth amino acid, between the fourth amino acids of each chain. So you can see here the nice helical rotation of the protein. Remember, the side chains are shown as green circles. They are more elaborate than what we are seeing at the moment. And here you can see the beautiful alpha helix that has formed with the primary structure. So if we now remove everything except the NCC backbone, you can see that the alpha helix forms a right-handed spiral that turns 360 degrees with every 3.6 amino acids. Okay, and the hydrogen bonds will be formed. So here we have the hydrogen bonds occur between the CO group of one amino acid and the NH group of another amino acid 4 units away. So it's not the adjacent one, but it's 4 amino acids away. The first amino acid, fourth amino acid, 8th amino acid, and so on. This is the hydrogen bond that gives us the alpha helical structure. So because they involve only the backbone chain reactions, they can be formed with many, many different side groups. So the side groups are not involved. The R groups are not involved in the formation of these hydrogen bonds. The R groups are all located towards the outside. Okay, so they're all sticking outside. So alpha helices can also spiral around each other, forming what is known as a coiled coil structure. So coiled coil proteins are also very commonly seen, and they are very often, they form with the help of hydrophobic reactions. So let me just go back a few seconds here and I'll pause it. So this, just a moment. So this hydrophobic interaction obviously would entail the formation of hydrophobic interactions between hydrophobic amino acids, right? So the hydrophobic side chains of the amino acids, such as tryptophan, such as tyrosine, phenylalanine, they would be located at this junction, which will allow 2 of these polypeptides, 2 of these alpha helical chains, which can be from the same protein. They most likely, more commonly, are from different polypeptides. They come close together to form what is known as a coiled coil structure. And this kind of a structure is seen very, very commonly in DNA binding proteins. A number of transcription factors that can go and bind to DNA actually have this sort of coiled coil structure. Let's have a look again. So this is how it looks. All right. Sharing my screen with you, let me just very quickly recap what we have learned so far. We have learned that the amino acid, the primary amino acid sequence, can have an alpha helical structure. And we will also see that the primary amino acid sequence can have a beta sheet, beta pleated structure, which we will see in a minute. And then we will learn about the tertiary structure and the quaternary structure. So what I would now like to revise with you very quickly is how the bonds are, what sort of chemical bonds are used to generate these higher order structures. Very often, these are weak bonds. They are weak bonds because they need to have the ability to be broken down and reformed. So weak bonds give you that freedom to break down the structure and reform it again. This breaking down and reforming of structure is not something that happens very dramatically in proteins, because protein structure has to be maintained for its function, right? So rather, small changes, small shifts in the protein structure can be accommodated. And these small shifts in the protein structure are called conformational changes that happen very frequently, and we will see that as well in a minute. So just to recap what kind of bonds are necessary for the formation of the higher order structure, we have hydrogen bonds that are formed in the, in the secondary structure, the alpha helix and the beta pleated sheet. We can have hydrogen bonds plus the hydrophobic interactions, plus van der Waals forces that can be found in the tertiary structure. And in the tertiary structure, we can also have disulfide bridges. Okay, so disulfide bridges are stronger bonds. They are covalent bonds. They are not weak interactions that we see in the hydrogen bonds. They are much stronger bonds, but they are necessary in order to keep the tertiary structure of the protein more stabilized. Okay, so stable structure formation requires the presence of slightly stronger bonds. And in this case, if the protein has a cysteine residue, then it can undergo a disulfide bridge formation. Okay? So now, if you're okay with it, I'm going to again go back to our Smart Biology book, and we will continue with the lecture.
Can I ask a question?
Yes.
Last week, I confused about something. Also, we speak about this in our WhatsApp group. We said NH and DO has hydrogen bond in it, right? Between and in a chemistry class, we said in our hydrogen bonds, some, for example, fluor, nitrogen, and hydrogen on it for hydrogen bonds. And also in not just intermolecular level, also intramolecular level also it is. But there is carbon, but still hydrogen bonds. Yeah.
So the hydrogen has to have hydrogen, right? The presence of the hydrogen bond invariably requires the presence of hydrogen in the structure. So as long as there is hydrogen which can attract electrons towards itself, and these electrons in this case, in the case of the specific case of the peptides, is provided by the oxygen. In the example that you were mentioning, most likely the electrons are provided by the fluorine, right? So hydrogen bond is a weak bond because it can be broken easily, and it basically involves the attraction of electrons coming from the oxygen towards the hydrogen. And this basically forms this temporary bond. But again, like I mentioned, if there are many hydrogen bonds, it becomes quite strong. So I don't know if that addresses your question, or are you talking about stronger bonds that may be formed within the structure?
I was confused about the chemistry rule. Fluor, nitrogen, and oxygen can have hydrogen bonds, but
so here ML is writing, and again, please help out here. My chemistry is not my strongest point. I think the hydrogen bonding occurs between the oxygen in the C double bond O in the carboxyl part and the H in the amino part. Yes, that's how it occurs. So it requires the oxygen and it requires the hydrogen. The oxygen is from the CO and the hydrogen is in the NH group. The CO and the NH that forms the hydrogen bond, keep this in mind, don't come from the same amino acid. In the case of the alpha helix, it comes from a neighboring amino acid that is about 4 amino acids away. But in this case, Specifically, the oxygen and the hydrogen is what is forming the bond. In the case of fluorine and hydrogen will form the bond. So it fits the chemistry. Oxygen is very, very electronegative. Hydrogen is present, and that is what is needed for hydrogen bonding, ML is saying. Yes, that is absolutely correct. So you need a highly electronegative ion, such as, or an atom, such as oxygen or fluorine, in order to form the hydrogen bonds.
Mm-hmm. Okay, thank you. I understand.
Okay. All right. Thank you, ML, for chipping in. It's really helpful. Okay, then I will go back to our.
I have another confusion
You
about.
have a question? Go on.
Yeah, I have another confusion about proline part.
Can you speak louder or write in the chat? I can't hear you, even though my microphone is completely at the maximum.
Oh, I'm sorry. Is it better now?
It's a little bit better, yes.
Normally, you have mentioned that proline doesn't have an NCC backbone. And if proline doesn't have an NCC backbone, how can it form a bond between another?
The proline has the carbon, and the proline has an imino group. So let me go back to the structure of proline. It will make it easier for us to understand. So it has the NCC. Do you see it? It does indeed have the NCC, but it also has this very unique sort of a ring-like structure, which is called an imino group. And this is what gives it the ability to form kinks.
Oh, so it does have an.
It does have. I mean, it wouldn't be classified as an amino acid otherwise if it did not have the NCC. All of them have to have the NCC to be classified as an amino acid that contributes towards protein synthesis. Because the R group of proline is a little bit different. It has a lot of CH2 bonds, right? And this will allow it disorders the peptide, the polypeptide chain, when proline is present. So while we are at it, I also wanted to show you this, although we won't be looking at the cysteine oxidation right now, but since we are in this slide, I wanted to show it to you as well. So these are cysteine residues, and cysteine is one of the three amino acids with unique properties: glycine, cysteine, proline. And you can see that cysteine is the only one that has the sulfhydryl group. Okay. And when we have the tertiary structure of proteins and the protein structure requires more stability, then it undergoes an oxidation reaction. So remember, removal of protons, removal of the hydrogen groups results in oxidation reaction, right? So the hydrogens are removed, and in this case, we have the formation of a disulfide bridge, and this is an oxidation process. And then this is reversible also in the presence of a reducing agent. In the laboratory, for example, we use an agent called beta-mercaptoethanol. This is a very strong reducing agent, which can actually reduce the disulfide bridges that are found in proteins and help them to become denatured. Okay, so we will talk about denaturation and renaturation of protein towards the end of the lecture. But just to give you an idea of how disulfide bridges are formed, I wanted you to have a look at this picture as well. The last question, was it addressed to your satisfaction? I can't remember who asked it, about proline.
Yes, thank you.
Okay, all right, great. So let's now move on to the secondary structure of the protein. So here we will learn about the secondary structure, and let's have a look at how it works. So the secondary structure here that we'll talk about are beta sheets. So beta sheets have a slightly more different shape. They are parallel to each other. So unlike alpha helices that have a spiral shape, beta sheets form these rigid, flat sheet-like structures. And they are held together again by hydrogen bonds between the NH of one chain and CO group from another chain, from the neighboring chain. Okay, so here we have the hydrogen bond here. This is the CO group, this is the NH group, and the hydrogen bond is formed between them. The green circles, as you know, are the side chains. So you can see this beautiful sort of sheet-like structure that can be formed as a result. So specific arrangement of atoms creates these pleats or folds in the sheet. And the side groups, they are extending outwards on both sides of the sheet. So when we put the side chains, then it kind of looks a little bit like this. So beta sheets can be formed from the same polypeptide that has folded in this manner, or from two different polypeptides. And because the NCC backbone, remember again what the NCC backbone is, because the NCC backbone of the polypeptide chain has a directionality, in other words, we have an N terminus and a C terminus, the beta sheets can be parallel or antiparallel depending on the relative orientation. So if two of the N termini that you can see, if you follow my cursor, you can see the two N termini are facing each other and the C termini are facing each other of the two polypeptide chains that are forming the beta sheet. In this case, they are parallel to each other, but if they are located in the opposite direction, then they would be anti-parallel. So let's have a look at how that is formed. These are parallel sheets. And then here we have some anti-parallel sheets where the C and N termini are located in opposite directions, and that's why they are called anti-parallel to each other. So if you can imagine a polypeptide chain, a single polypeptide chain, going in this manner and then folding back upon itself, you can expect that chain to be anti-parallel. So we start with the N terminus, we move, we have a fold, and in this fold we may have a proline residue which causes it to fold, and then we come back again to the C terminus. Okay, so in this case it would always be anti-parallel to each other. And while we are in this picture, this really, really beautiful picture, let's try to understand where we can have beta sheets. What type of proteins tend to have beta sheets? Beta sheets, I don't know if you recall from the lecture yesterday of the shape of cellulose. Cellulose protein, cellulose, the complex carbohydrate, has had this shape of parallel chains of glucose molecules, parallel chains of the polymerized glucose molecules to each other. This sort of a parallel structure of any polymer gives it a very, very high amount of structural support, gives it strength. Okay, so beta sheets are generally insoluble. Proteins that have a lot of beta sheets tend to be highly insoluble. They tend to have functions that are related to structural support. So for example, silk proteins. Silk proteins have very high proportion of beta sheets in them. A lot of our extracellular matrix proteins have very high amounts of beta sheets in them. Generally, beta sheet containing proteins that have very high amount of beta sheets are insoluble in nature, whereas proteins that are highly soluble in the cytoplasm of the cell tend to have more alpha helices in them. Okay, there are proteins like myoglobin, if I'm not remembering incorrectly. Myoglobin does not have any beta sheet at all. So there are proteins that have no beta sheets, and there are proteins that have a lot of beta sheets as well. And depending on the presence of the beta sheet, the structure of the protein can be predicted. If the beta sheet component of a protein increases, then the protein can become insoluble. So this is an example that we read, that we give every year on prion proteins. So prion proteins are proteins that were discovered in the 1980s to cause a disease that was called mad cow disease. Okay, so this mad cow disease started in England with cattle, with cows that could not walk in a straight line, and so locally it was called that the cows have gone mad. And it turned out that the mad cow disease was not caused by a virus, which is what was initially suspected. It was not caused by a virus, but rather it is in fact, it's not caused by a microorganism at all, because several experiments were done in order to confirm that. Rather, it was found that these proteins called prion proteins that exist in our brain, they have specific functions for metal binding in our brains, they changed their secondary structure in such a way that they had much, much more beta sheets in the structure than what they had before. And this presence of beta sheets, increased beta sheets in the structure, caused these prion proteins to precipitate in the brain, okay, in the brain parenchyma, which means just inside or out, more commonly outside of the neurons. And this resulted in what is known as a spongiform encephalopathy. So the brain, after the animal died, looked like a sponge, okay, when the autopsy was carried out. So it had many holes in it. In the mid-1980s, there were incidents of humans who started developing this disease. And it turned out that all of these humans ate beef, ate meat from those animals that had developed mad cow disease. So this was very, very interesting. And again, at that time, the hypothesis, of course, was that this is an infectious agent that comes from the beef to the human being through the food. But then again, experiments were carried out, and it turned out that there is no microorganism in this in the beef. Rather, the meat contained these prion proteins from the animal that has had the disease, and these prion proteins, when it gets into the digestive system, they are very, very resistant to proteases that we have in our digestive system. They get into the blood, they go through the blood, cross the blood-brain barrier, get into the brain, and then when they come in contact with the human prion proteins, the diseased prion proteins change the secondary structure of the human prion proteins, increase the beta sheet. So it's kind of influencing the prion proteins of the host. And then these prion proteins now have increased beta sheets, and they precipitate, causing what is known as CJD, Creutzfeldt-Jakob syndrome disease. So in this case, the humans developed mad cow disease, and they died. There is no cure for this. So many humans died, and at that time in the mid-1980s, the cattle industry in Britain had a major fallout because of this. Many people went bankrupt, and many farms had to be closed down because all the animals had to be culled, had to be killed. Okay? So in this case, remember, there is no change in the amino acid sequence, in the primary sequence of the protein, right? There is a change in the secondary sequence. So this does not involve any mutations. It involves a change in the hydrogen bond formation, which is seen in the secondary structure of the protein. I will give you a chance to ask me questions once we go through these videos. Okay, so polypeptide chains can also adopt other secondary structures, so such as disordered loops and flexible chains. So here we have some disordered loops. Here you can see the helical shape, the alpha helix here, but this is a disordered loop. And in the slide that I showed you from the PowerPoint, you also saw that there were some beta sheets, there were some alpha helices, and then there were some loops. And in the kinks of the loops, which we can see in this edge here, most likely we have proline residues, which allows this protein to fold back on itself and make these disordered structures. Okay, so this is a disordered loop. Here we have some flexible chains. So this is how the secondary structure is. So it's not all alpha helix and beta sheet, rather there can be other structures as well. I think there is a question. After disulfide bridges, after disulfide bonds are formed, wouldn't the extra 2 hydrogen, oh, no, no, it wouldn't disrupt the pH level in the cell, because number one is those hydrogens would be immediate, this is a good question, they would be immediately mopped up by a hydroxyl group somewhere. So we have a very good buffering system found in our cells. Plus we don't have that many hydrogen ions being produced. There are not that many cysteine bonds being formed, disulfide bridges being formed, that it would change, it would overwhelm the buffering system of the cell. So, but we do have a buffering system in our cells to mop up these extra hydrogen ions that are formed. Okay, now let's come to the tertiary and quaternary structure of the proteins. And again, we have some really, really nice videos here. So let's have a look. So secondary structure elements, the alpha helices and beta sheets, they will then come together, as we saw in some other pictures as well, to form the domains. So proteins generally have multiple domains, as we were talking about earlier during the lecture, and they have flexible linker regions. These linker regions generally don't have an alpha helix or a beta pleated ordered structure. They are more disordered. And in some instances, a single alpha helix could also represent a domain. For example, if you have a transmembrane protein, then this transmembrane protein can have a helical structure, an alpha helix, as the membrane binding domain. Okay. Remember, the outer amino acids of the helix will have the hydrophobic amino acids. So there are some questions. Let me see. Why do too many beta sheets cause the brain to be spongy? Plaques which kill the neuron. Yeah, exactly. So the neurons are there dying, and basically it looks like a sponge. It does not really, yeah, exactly. Your friends have answered the question for you. Thank you. Okay, so let's proceed. So here we have a protein. So this, look at this, oh, it's so beautiful. This is a protein that is a membrane associated protein. So it's not, a small part of it is going into the protein, but a lot of it here, for example, this is associated with the hydrophobic, with the head regions, the hydrophilic head regions of the membrane. Okay, so these are membrane associated proteins. So this is a flexible linker, again, disordered structure. And here we have a membrane-bound protein where we have the alpha helix that is located within the membrane. So different structural domains are responsible for different functions, and each domain has its own compact three-dimensional structure. So there might be a membrane embedding domain, there may be a DNA binding domain, and here we therefore come to what is known as the tertiary structure. So all these domains together form the tertiary structure of the protein. It's the three-dimensional folding of the entire polypeptide chain, and the tertiary structure is held together by the same types of non-covalent bonds that are found in the secondary structures. Secondary structures were primarily formed from hydrogen bonds, right? In the tertiary structure, we also can have hydrophobic interactions. We can have disulfide bridges, which are slightly stronger, much stronger actually, they are covalent bonds, but mostly many proteins actually don't have cysteine residues in them. In that case, the tertiary structure is primarily formed by non-covalent bonds and interactions. So usually, as we have been discussing, the hydrophobic residues are found to the interior of the protein, while the hydrophilic residues are exposed to the outside. And again, there are proteins that are formed of only a single polypeptide chain. This will be the end of the organizational hierarchy. In other words, this is the final form of the protein. The protein cannot form any more higher order structure, and therefore now this is called a protein rather than a polypeptide. Okay, it's referred to as a fully formed protein. So you can see here the hydrophilic residues. Let me just go back a few seconds. You get so you can see here all the hydrophobic, the groups that make it hydrophobic are located towards the inside, and the OH groups and the hydrogens were all located towards the outside. So that's what gives hydrophilicity to the protein. So many proteins are composed of multiple interacting polypeptide chains. So we were discussing this during the break of how a highly complex protein may be formed of more than one polypeptide chain. And it was rightly pointed out by your colleague that it seems difficult to understand how such a complex protein, such as the pyruvate dehydrogenase that we see in the left corner here, how can it be formed with just one polypeptide chain? It just seems impossible. And you're right. There are many proteins that are formed of multiple polypeptide chains that are synthesized from different mRNAs, even completely different mRNAs, and they can come together and they can form what is known as a quaternary structure. So the quaternary structure will have several folded polypeptide chains. They are folded first and then they come together to form what is known as the quaternary structure. Sometimes it's called a protein or it's called a protein complex. In the case of pyruvate dehydrogenase, it's called a complex. So here we have several polypeptide chains. It's already folded and then it will come together in order to form what is known as the quaternary structure of the protein. So we have various subunits. Again, they may not have any domains, right? So remember difference between subunit and domain. Subunits are different parts of the protein, but they may not have a specific DNA functional domain in them. So a domain usually is associated with function. A subunit may or may not be associated with a function. So here we have a protein/protein complex. So we learn a little bit more about quaternary structures here. And so protein subunits are held by the same type of covalent, non-covalent bonds. So we can have, again, hydrogen bonds, we can have hydrophobic interactions, we can have van der Waals forces. In many proteins, hydrophobic interactions play a very, very important role. So the hydrophobic amino acids are located towards the interior of the protein, remember. So hydrophobic interactions therefore become very, very important in bringing these subunits together. So some protein complexes contain a small number of subunits. For example, this is a dimer. This dimer is triose phosphate isomerase. This is a protein found in glycolysis. Whereas there can be a tetramer, for example, glyceraldehyde 3-phosphate dehydrogenase, again, a very, very important protein in glycolysis. This is a tetramer of 4 proteins, 4 subunits that come together to form this complex. Other protein complexes can have dozens of subunits forming huge machines. So here we have 2 subunits, 4 subunits, and then we have the proteasome, which has 64 subunits, and we have the ATP synthase, which has 56 subunits, and our favorite pyruvate dehydrogenase, which has 102 subunits. So it's amazing. 102 peptides that fold and come together forms the pyruvate dehydrogenase. And the interesting thing is the pyruvate dehydrogenase in bacteria look very, very similar. So these are the different proteins that we have come across with their tertiary structures. So the non-covalent bonds and interactions that hold together all secondary, tertiary, and quaternary structures, they allow for quite a bit of flexibility. So that's what I was trying to indicate earlier on, that if we had a lot of covalent bonds, then the protein structure would not have the flexibility that sometimes is needed for its function. Covalent bonds give structure, it gives stability to the structure. So let's have a look at the disulfide bridge now. So sometimes covalent bonds are added to staple certain regions together. So this brings them very close together and gives a lot of structural support. So proteins in the extracellular environment, in the extracellular matrix, are usually stabilized by such covalent cross-linkages. So here we have covalent interactions, which is most likely formed by disulfide bridges. And these extracellular matrix proteins, for example, include fibronectin, it can include laminin. These are all proteins that have a lot of disulfide bridges in them. Okay? And it gives them a lot of stability, which is needed for the extracellular matrix, because the extracellular matrix needs to be strong. So they can occur within the same polypeptide chain, or they can occur between the different subunits of this protein. They typically don't change the conformation of the protein, but they reinforce the existing shape of the protein. So it doesn't change the shape of the protein. So the most common of these is the disulfide bridge, the disulfide bond, which is formed between sulfur atoms of two cysteine amino acids. And earlier in the lecture, you saw how this disulfide bridge is formed by an oxidation reaction, and then it can be deformed by a reduction reaction. They're usually not found in cytosolic proteins, because this is very, very interesting, because we have a reducing environment in the cytosol, okay? Which means there are many reducing agents, there are many electron donors in the cytoplasm, which will potentially break these disulfide bridges. So very, very rarely do we have disulfide bridges in soluble proteins in the cytoplasm, because the disulfide bridges cannot be maintained. So disulfide bridges are found much, much more in the extracellular environment and in membrane-bound proteins that face the extracellular environment in order to give them a structural support that is very, very important for their function. So these are the cysteines. We will have loss of hydrogens, and then we will have a disulfide bridge being formed. So there are two more videos that I will share with you, and then I will open up for some questions. So what sort of binding affinity and conformational changes have we been talking about? So just, for the few minutes before this video, we were talking about how we need to have some amount of flexibility in the structure of the protein. So what we will do now is try to understand why we need to have some amount of flexibility in the structure of the protein. So here we have a phosphorylation event whereby a kinase will come and bring the phosphate, an ATP molecule, and the phosphate will be added to a substrate. A kinase is an enzyme which will take the phosphate group from the ATP, and it will add that phosphate group to a receiving amino acid. As I mentioned at the beginning of the lecture, there are 3 amino acids that can receive phosphate groups, and they are tyrosine, serine, threonine, and tyrosine. So here we have a kinase, here we have G protein coupled activation, beautiful, beautiful, beautiful pictures, where we have the activation of adenylyl cyclase. We've also seen this before, where we have the formation of the actin filaments, and then we have a large number of proteins. The proteins can randomly collide with other proteins because it's very, very crowded inside the cell environment, right? We saw this last week as well. So randomly, proteins will collide with each other, but only those proteins that have surfaces with the amino acid distribution, which are compatible with another protein, will they come close enough to each other. In general, proteins don't come very close to each other, but sometimes they do if they have compatible surfaces, okay? And they form these non-covalent bonds or interactions, which will keep them together for the duration that they need to be together. So the chance of the 2 proteins remaining bound to each other will depend on the very, very precise 3-dimensional arrangement of their atoms. If it favors it, it will remain bound. If it does not favor it, it will be a kiss and tell, kiss and go. So it will just join and then it will go away. So here we will have a look at what we mean by compatibility. And for this, we are going to be focusing again on hydrogen bonds, okay? So we have protein 1, which has a side chain like this, protein 2, and again, these 2 can come very close to each other and they can form an interaction. But if the arrangement of the side chains is not compatible, then they will not come close enough to each other to form any kind of non-covalent bond which will allow them to interact with each other, neither hydrophobic interactions nor hydrogen bonds. So here we have low affinity interactions, and so they don't come very close to each other and they wouldn't form a complex with each other. When the affinity is high and these interactions occur, the non-covalent bonds will and may cause some shift in the position, in the structure of the protein. So because these amino acids are covalently connected to other amino acids, so we are talking about the peptide bond here, each amino acid is covalently joined to the next amino acid. We learned that at the start of the lecture. This movement can cause a shift in the secondary structure, right? So the hydrogen bonds between, so the alpha helix structure or the beta sheet structure, or even if it's not alpha helix or beta sheet, any secondary structure that is formed will be shifted, and the tertiary structure will be shifted as well. And again, I will repeat myself, these shifts are possible because of the non-covalent bonds. Covalent bonds are too strong to undergo any shift. So it's the hydrogen bonds and the hydrophobic interactions that undergo primarily the shifts. So this will cause movement of changes in the secondary structure as well. And if the secondary structure changes, sometimes the tertiary structure can change as well. So now the protein is said to undergo a conformational change. This is a word that you will come across very, very often, a conformational change, which is basically a change in the shape of the cell, of the protein, which will change the function of the protein. It will open up new functions of the protein, perhaps because of this change. So let's have a look at what conformational change there is. So here is a protein, which, let's go back a few seconds. This is a protein that, did you see? It underwent a conformational change. So here we go, a major conformational change, which will give this protein new functions, perhaps. I will now, I think there are some questions. Let's have a look at what the questions are. So do we know if the formation of protein complexes happened accidentally in the evolutionary process and functioned better and so have remained as it was, or was it inevitable because of their 3D shape? Was the combination on purpose? Good, very good question. I think it's a combination of both, because there have been major movements of blocks of nucleotides from one region of the genome to the other. So this has resulted in the formation of proteins with unique structures, but common structures, right? So if there is a copy, like I was mentioning earlier, that there are domains that are found in common between completely unrelated proteins. So these common domains will allow the proteins to interact with each other and to interact with other macromolecules. So sometimes it's accidental because of the way in which the mutation or the translocation event worked. In some case, it may be inevitable because the proteins are around each other. They are available to each other, and therefore it was inevitable that they would go and bind to each other. So I think it's a combination of both. And then there's another question on the compatibility of the molecules, like the interactions between them. Gizem, you're welcome. So here, the compatibility of the molecules come from the specific arrangement of the side chains of the amino acids, as well as the location of the NH and the CO group with, well, mostly the side chains. I think when I'm trying to answer this question and I'm picturizing how two proteins can come close to each other, what comes close to each other are the side groups, because the side groups are mostly towards the outside. So I would think that the compatibility comes from the composition of the side chains of the amino acids. And if the side chains are compatible, in other words, if the side chains have the hydrogens and the oxygens in the correct position, or the CH2 groups in the correct position, that will allow them to come very close to each other via hydrogen bonds in the first instance, or hydrophobic interactions, or van der Waals forces in the next instance. Then it will result in compatibility. These molecules are compatible with each other. But if there is a section of the protein where we have some hydrophobic amino acids and hydrophobic side chains, and the neighboring protein has hydrophilic side chains, then they are not compatible with each other. Or if they have hydrophilic side chains, but the hydrogens and the oxygens cannot come close enough to each other to form the hydrogen bonds, then they are not compatible with each other. So overall, the 3D shape defines whether they can come close enough to each other to form the specific non-covalent interaction. And that's what is defined by compatibility. Is that good? Okay, good. Right, so now we will come to the last video that we will watch today. And that is, again, a continuation of the binding. And here we will specifically talk about a protein called protein kinase A. For protein kinase A, this is a very important signaling molecule, which is also a kinase. So a kinase is an enzyme that is capable of taking the phosphate from ATP and transferring that phosphate to a substrate protein. Okay, and this substrate protein, again, I'm repeating myself, but the substrate protein will have to have an amino acid with a hydroxyl group, such as the serine or the tyrosine or the tryptophan, which can then accept the phosphate group, serine, tyrosine, and threonine, not tryptophan, serine, tyrosine, and threonine. These are the 3 amino acids that have the hydroxyl group. It can receive the phosphate group and it can get phosphorylated. So a kinase phosphorylates a protein substrate by taking the phosphate from ATP. Now, protein kinase A is activated in the presence of a small molecule called cyclic AMP. And this cyclic AMP is produced, and I will ask you to recall the very beautiful G protein activation video that we saw a few minutes ago, where the G protein shifted after it was activated by the G protein coupled receptor, and it combined with another membrane-bound protein. That membrane-bound protein is adenylyl cyclase, which converts ATP to cyclic AMP. And this cyclic AMP will now bind to protein kinase A and activate it. So these kinases are generally very, very, very active molecules because they can phosphorylate substrates, right? And this can cause major changes in signaling in the cell. So these are highly regulated. And in the case of protein kinase A, what we will learn is that it has a regulatory subunit which keeps the protein kinase A inactive until the cyclic AMP can come and bind and activate it. So here we will learn about how protein complexes can also have regulatory subunits in them. So let's have a look. Okay, so conformational changes, as we were seeing in the previous slide, it leads to different 3-dimensional shapes, and it can basically change the way in which a protein can interact with other protein. So now that this protein has undergone a conformational change, it can interact with a protein that it would not be able to interact with otherwise. So pKa is composed of 2 regulatory subunits and 2 catalytic subunits. So these are the regulatory subunits, and then we have the catalytic subunits, which are in orange and in red. In the absence of cyclic AMP, the regulatory subunits hide the catalytic subunits, and it prevents this protein kinase A from its activity. It's an inactive enzyme at the moment in the absence of cyclic AMP. So it conceals the critical amino acids at the catalytic subunit, preventing the protein from carrying out its functions. So this spelling is wrong. The critical amino acids are concealed here. And as the levels of cyclic AMP rise because of signaling that we saw earlier, now cyclic AMP has come. They bind to the regulatory subunits, and this results in a major conformational change. So you can see the conformational change now. So that's pretty amazing. And as this rearrangement happens, it results in a change in the tertiary structure. Again, these sort of changes are possible only because we have non-covalent bonds. And it repositions key amino acids in such a way that it significantly decreases their binding affinity for the catalytic subunit. So now the regulatory subunits have moved away. Look at that. So they have actually moved away, and now the protein can be functional. So for actin polymerization as well, again, we can have similar interactions. For transcription elongation, so this is protein that is working during transcription. For protein kinase A activation, in each case, we are seeing major changes in the conformation of the protein, which will allow it to function when it's needed. Okay, so with this,